Okay, so today we are taking a look at the past paper questions coming from the work in chapter 5, which is our enzymes. Okay, let's dive into this long question. Protein digestion begins in the stomach of the human alimentary canal and is completed in the small intestine. Describe in detail how enzymes function to digest protein in the alimentary canal. This question is for 6 marks. And it basically encompasses the whole process of how enzymes function to digest proteins. In detail, tells us that they want everything on how enzymes function to digest proteins in the alimentary canal. Well, enzymes function as biological catalysts. Many chemical reactions can be speeded up by enzymes, and enzymes also lower the activation energy of these reactions. It's also important to mention the lock and key mechanism, so you can refer to how the shape of enzymes and the shape of the substrates are complementary to one another. And then also the active site, which is the site where the substrate fits into the active site of the enzyme, and this forms our enzyme substrate complex. It is also important to mention that enzymes are unchanged in this reaction, as well as that enzymes split proteins into their precursor molecules, amino acids. You can refer to pepsin, which is active in the stomach, and trypsin, which is active in our small intestines. For this question, I would suggest to go and write out the whole process, as well as all the properties of enzymes, and this will help you prepare for any questions relating to the questions in chapter 5. So you should probably be aware that some people are unable to digest lactose, milk sugar, and have a condition known as lactose intolerance. These days, more and more people are struggling from this. Figure 1.1 shows what happens in the intestine of a person who is lactose intolerant if they eat food containing a lot of lactose. So basically, this is from the stomach, and over here food travels, and up here gets mixed with water. Bacteria to digest lactose to simple sugars are found over here. Bacteria to produce hydrogen, methane, carbon dioxide. Build up of gas, small intestine, large intestine, and the result is diarrhea and gas, because as you can see, the lactose is not being broken down. So we asked to explain why lactose is not absorbed by the small intestine. And the reason why it's not absorbed is because the molecule is too large to be absorbed if it's not broken down. And because the enzyme to break it down is not present, the molecule just travels through the GIT and is not broken down at all. So let's take a look here. Many enzymes do not work at temperatures above 60 degrees Celsius. Which statements explain this? Well, most enzymes are denatured at high temperatures, which basically means that they can't function because their shape has been altered. So we need to look for a statement which states that, maybe in different words. Product molecules are not made because the active site has changed shape. Right off the bat, that sounds correct. But before we choose that as our answer, product molecules change shape, so they do not fit the active site. No, the product molecules do not change shape. That's incorrect. Substrate molecules are moving too fast. That's also incorrect, because if substrate molecules were moving faster, that would actually increase the rate of the reaction. Then substrate molecules are moving too slow. That is also incorrect, because at 60 degrees Celsius, substrate molecules will not move too slow, they'd actually move quite fast. So our answer here would be A. So let's take a look here. This table shows the enzyme activity of alcohol dehydrogenase at different temperatures. At 30 degrees Celsius, there's a activity of 105 arbitrary units. At 42 degrees Celsius, there's an enzymatic activity of 175. So we can see with the increase in temperature, there's an increase in activity of our enzyme. The information in table 5.1 shows an increase. That's just basically exactly what I just said. And then explain why an increase in temperature causes an increase in enzyme activity. Well, we've learned that if there's an increase in temperature, that this will actually increase the amount of kinetic energy of the molecules moving around. This will result in faster moving molecules, which will result in more collisions between substrate molecules and our enzymes. This will result in more substrates binding to the enzyme's active site, so more successful collisions, and this is why with the increase in temperature there's an increase in enzymatic activity. So state one factor other than temperature that affects enzyme activity. Well, that would be pH. pH affects the activity of our enzymes. Certain enzymes function better at high pHs and other better at low pHs, and then there's a whole bunch of them that function optimal at a neutral pH. So lactase is a human enzyme that catalyzes the breakdown of lactose in milk. At what temperature does lactase work fastest? Well, it's a human enzyme, which means that it's inside the human body, which means that it works optimal at our body temperature, and the closest one here would be 37 degrees Celsius. So over here we get shown a graph which shows the effect of pH on a particular enzyme controlled reaction. This is our rate of reaction, our pH is over here. So immediately I can see that the rate of reaction is highest around pH 7, maybe 7.5, and over here there's a decrease in the rate of reaction to one where there's probably no rates. There's the rate of reaction equals 0, and here it also decreases and the rate of reaction reaches 0 at 13. So the question asks, when is the enzyme not active? Well, we basically just answered that, and we can easily see it on our graph here. Over here we're looking at rate of reaction. Where is it 0? It's 0 at 1 and 0 at 13. So I hope you enjoyed today's lesson. Good luck with the studying and go and get those good marks.